what is sequence alignment. Now, the previous module was to show you why, exactly why, um, to keep at the back of your mind the DNA structure or the RNA structure while you just took it for granted and wrote a sequence out as a primary sequence. What is sequence alignment? So sequence alignment is a way of arranging the primary structure. And when you talk about the primary structure, we're talking about the sequence. No secondary structure. We're not worried about bonds. We're not worried about the three-dimensional nature of it. Of DNA, RNA, or proteins, such that the similar regions are aligned with each other. Well, why do we need to do this? Because of consequences to structure, function, and evolution. To see why did a DNA sequence change, and has the change in that sequence, is that change in the sequence sufficient to merit calling it DNA for a new species? Okay, we'll discuss a little bit very briefly about speciation. You want to take two sequences and you want to align them to find the differences and similarities. Are these two things that are two entities in a DNA sequence that are aligned? Are they identical to each other? Are they similar to each other? And in course of evolution, we begin to see how one parent species uh, evolved into daughter species, which further evolved into daughter species, and further evolved into daughter species to give rise to several different species. The orthologs, homologs, and paralogs are what I allow us to begin to understand this. Now, in terms of sequence alignment, we do it as a matter of course now. There's so many software which allow us to do some of this. But I'll give you one specific example, and it happens to be in my area of research. Um, for example, if you ask yourself, well, why do we need sequence alignment? Okay, so a case for sequence similarity for the organization of genes into families and subfamilies. Now, olfactory receptors, and these are sequences that align the mucous membrane of our nostrils, they interact with molecules of smell coming in from the outside, and they attract the olfactory processing part of the brain, which is about here, and so you'll see that when an olfactory receptor interacts with an odorant, a molecule of smell, the signal is passed to the olfactory processing part of the brain, and then that results in our perception of smell. Okay, is it a good smell? Is it a bad smell? Is it some smell which we have to stay away from because it's necessarily dangerous to us? Now, the interesting part about olfactory receptors, in addition to their function, is that they are the largest family in the human genome, about 20% of the genome. And they found that it's the largest family in rats and mice and dogs and even elephants, which has 3 times the number of genes. So several groups, once the human genome came out, they went out and extracted putative olfactory receptor genes, potential olfactory receptor genes, and they found there was about 1,000. But there's about, give or take, depending on whom you ask, 400 functional genes, and the rest of them are pseudogenes. Now, you could make various cases for why we started out with 1,000, and somewhere along the line, some of these were rendered non-functional. You could say, for example, well, because we have other faculties, right? We are sentient. We can think. We can reason, and we can see. We can hear. We have all senses. They're not perfect, but we have several senses working at the same time. Because remember, all these senses are designed to survive, right? Flight or fight, and what kind of reaction are we going to have? Because if you look at the functional genes in rat, mouse, dog, etc., you'll find a lot more functional genes in these lesser species, where they have to rely more on their sense of smell. At least this is one theory. There's several competing theories as to why we currently have about 400 functional genes out of 1,000. Now, if you consider 400 out of 20,000, that's 1,000 out of 20,000. That's a pretty big number, okay, for genes. So when people extracted this information from the human genome, they said, how do we organize this large family of genes? This is not one gene which does one or two or three different functions. So they did a sequence alignment, a multiple sequence alignment, where they lined up and sequenced, and aligned all one with everything else and one with everything else, and then they grouped these into based on similarity. So they said, if this is greater than 40% similarity, we'll call this group a family. Now, this is arbitrary. It's arbitrary in the sense that you could say, well, why 40? Why not 30? Why not 20? Why not 45? Why not 50? But it might be arbitrary, but you would say, well, it's reasonable. And then if groups that had more than 40% sequence similarity, they were considered families. And within those families, if these sequences had 60% similarity, 60% or greater, they were called subfamilies. Now, this work was, now several different people, as I said, identified these sequences, and they each group called it something. Each group, they had their own system of organization. They did agree in most part. There were some disagreements, but this particular criterion for determining what categorizing based on families and subfamilies came at the Weizmann Institute of Science. So in terms of families, they just gave them numbers, 1, 2, 3, 4, etc. And subfamilies, they just gave them letters. So for example, if you had an olfactory receptor which was called, let's say, 1AE3, that means it belonged to family 1, it belonged to subfamily A, and 3 was just a number assigned. This was the third member of the A subfamily in family 1. Okay, and so alignment was important. This is one example where alignment was important in order to be able to categorize. Alignment is also important to understand evolution, as we'll see in this slide. So very often when we think in terms of, are two sequences identical? Are they similar? Very often this is used loosely, very often inaccurately, as, oh, this is homologous. Oh, I see. I'm measuring the sequence homology. But that's not necessary, because when we talk about homology, we often talk in terms of, well, it's a sequence identity. Is it a sequence similarity? But that's not the proper definition, right? So genes related to a second gene by descent. Now, when you say related, it means similar. If you kind of match them up, they are similar by descent from a common ancestral DNA sequence. So that is what is called a homologue. Okay, that they are comparable, but they have to have a common ancestor. Now, a homologue may apply to a relationship between genes that, as I said, evolved, became slightly divergent because of speciation. What is speciation? The formation of a new and distinct species during evolution, or to the relationship between genes separated by events of genetic duplication. So, for example, here you'll see the chromosome, and just an example of a chromosome, and this region EF is duplicated. And the moment it is duplicated, it then becomes different. Okay, and so what you'll see here is that it's a chromosome within the same species which is different, and in the case of speciation, it forms a different compound. Let's look at orthologs. So orthologs are genes which are in different species but evolved from a common ancestral gene. So you see that speciation gives rise to orthologs. Okay, and there is a picture in this slide at the end which will explain this in greater detail. Well, what is a paralog? A paralog is a gene related by duplication within a genome, which means these are two sequences which have some similarity. The function is necessarily diverged, but they belong to the same species. So ortholog, different species; paralog, same species. So you can also think in terms of a paralog being generated by, one example would be genetic duplication. Within the same chromosome, you have something different, which translates into something different, into a different function, but the species is the same. Now let's look at this overall picture. So homologue is kind of a loose, but it's an umbrella definition. So look at it. Let's look at it top down and then bottom up. Okay, for the top down is for the definition. So we have an ortholog. So this is an ortholog. So you say that the alpha gene or the alpha protein, or a protein associated with a specific function, there is similarity between frog, chick, and mouse, but So the function is the same, the sequences are likely similar, but they belong to different species. Okay, so speciation. So let's look at it top down and then bottom up. Similarly, here you have another function, another protein, another gene, which is in different species. So, but they are similar in function and likely similar in sequence, or identical, or very close in sequence. So these are orthologs. Now, two different genes, two different functions, but within the same species, probably due to genetic gene duplication, results in paralogs. So orthologs, different species; paralogs, same species; orthologs, same function; paralogs, different function. Now let's look at it from the bottom up. Let's say you had a globin gene, which did something, right? And within a particular species, gene duplication resulted in some change, and that resulted in two separate genes, resulting in two separate functions. Now, this gene from a particular parent species gave result via speciation, three different species. Similarly here, okay? But the function is retained. And therefore these are orthologs. Okay, so that explanation, I hope, will give you some sense of orthologs, paralogs, and make sure that it's not, not interchangeably used or not loosely defined, or homology is not spoken of synonymously with identity or similarity. Remember, for homology to occur, you have to have a common ancestor. One more thing to mention, you notice that you see this chain. So frogs belong to amphibians, chicken belong to aves, and mouse belongs to mammals. So you see that the frog is a lot more primitive because this species diverged a lot earlier in the evolutionary chain, and the chicken and mouse, the birds and the mammals, diverged a lot later, right? So if you think in terms of higher order species, the fact that this line for chicken is not just a three-pronged like a fork, but it occurs here, it gives you some idea of when that break occurred. So what is a little more higher ordered species, right? It's important to begin to understand that the way the branching occurs actually means something. And when we learn about phylogenetic tree and cladistics and what happens during alignment, okay, why do things change? Why do things modify? Why do things, in addition to in addition to gene duplication, what else occurs? So there are mutations which occur, which means a nucleotide changes. Consequently, the amino acid changes. Consequently, the protein that's the gene translates into changes. Consequently, the function changes. And if there's significant changes in the genome at a genome level, then you have speciation. So insertion of one or more nucleotide base pairs into a DNA sequence. So you insert something or you take something out, and that's really important when you begin to think in terms of speciation. So deletion is where one or more nucleotide base pairs or an entire region of the DNA is missing. This is often insertion deletions are combined to be called indels. So let's say you are aligning two species, right? And you align them and you say, well, okay, so far so good, but if we want this P to align, you're going to have to put a gap in here. Okay, you're going to put a gap. So this is what is called a gap. And then you say, here's the result of my alignment, exactly the same. And when there is a mismatch, you just put an X. Okay, and typically that's convention. And here there's a gap, so you put a gap. Now, very important to understand this. Does nature put gaps? No, it doesn't, right? Nature has a sequence, which is a sequence, and whatever sequence structure, functional interactions and consequences are a result of nature. Things like gaps and deletions, things that we introduced, not deletions, but gaps. We introduced gaps during an alignment so we can understand what goes on, okay? Nature doesn't say, I'm just going to have nothing. There is no such thing as nothing, right? This is something we understand, so we can understand alignment, because because once we understand the alignment, we can understand, categorization of families, subfamilies. We can understand the evolutionary consequences of why a change occurred. Okay, so remember, a gap is a human construct. A gap is a human construct. Okay, a gap doesn't occur in nature. There's no such thing as a missing amino acid in nature.